really excited to be here to present this primer topic, Introduction to Pharmacogenetics, having attended MPG primer for several years now. I have no disclosures today. Here is the outline of the talk. So we'll start off with some general principles and nomenclature relevant to pharmacogenetics. I'll then dive into different examples of sources of pharmacogenetic variation, discuss how pharmacogenetics has been applied to type 2 diabetes, discuss some resources for those who are interested in pursuing this area, as well as discuss challenges and future directions in the field. So to start, a little bit of background on adverse drug reactions. Unfortunately, in the United States, 6.7% of hospitalized patients have a serious adverse drug reaction and responsible for 100,000 deaths per year in the United States. These reactions are associated with prolonged length of stay, increased costs of hospitalization, and increased risks of death. And in one study looking at emergency department visits for these drug reactions in individuals who are elderly, greater than 65 years of age, 60% of these were related to drug reactions in 3 distinct classes, and not surprisingly, anticoagulants, which are blood thinners, anti-diabetes agents, causing hypoglycemia or low blood sugars, and then the third class is opioid analgesics. There are many factors that influence drug response, both intrinsic and extrinsic. But for the purposes of this talk, we'll specifically focus on pharmacogenetics, which is the study of genetic variation, influencing individual differences in the therapeutic or toxic response to a drug. The goal of pharmacogenetics is really to change the approach from one drug fits all to one where we personalize the approach, and patients based on their genetic variation can be given the optimal drug and dose to maximize efficacy and reduce toxicity. And so this may mean an individual receives a higher dose of a medication, a lower dose, or an alternative medication altogether. I'd like to, at this point, clarify key terminology in this area that'll be used throughout the talk. And so we use the term pharmacogenetics as well as the term pharmacogenomics, often interchangeably. Pharmacogenetics traditionally has referred to single gene drug interactions, whereas pharmacogenomics generally refer to studies that utilize genome-wide approaches, which can incorporate other elements such as epigenetics. When studying drug action in individuals, we focus on 2 major determinants, pharmacokinetics and pharmacodynamics. Pharmacokinetics refers to how the body processes a drug, and that can range from initial absorption of the agent to distribution in the body, metabolism of the drug, as well as excretion and disposition from the body. Pharmacodynamics refers to how the drug acts on the body, and basically that is related to how well the target cells in the body, the tissues or receptors are responding to the drug. And both pharmacokinetics and pharmacodynamics are critical considerations in the field of pharmacogenomics. I'd like to, at this point, kind of review some nomenclature conventions. And so all of you are familiar with the use of the reference SNP number to identify genetic variants. But in pharmacogenetics, there's also the STAR nomenclature. And so certain gene variants can lead to altered function of the encoded protein. And this varies based on the gene of interest. And so this nomenclature has been developed to kind of reflect changes in activity due to polymorphism. So the STAR1 allele typically refers to normal activity or wild type, whereas STAR2, depending on the gene, could be decreased enzymatic activity, increased activity, or no activity, depending on the specific gene. And you can't infer really that STAR2 is in one direction or another. It really depends on the gene you're interested in. The STAR nomenclature can be used to describe genotype as well. And so, for example, homozygous carriers of a STAR2 allele in a given gene are denoted as STAR2, STAR2. We can also define haplotypes or set of linked SNPs throughout a gene. And so this can be a combination of alleles or a set of alleles and across different positions that may have a specific functional effect. Not all drugs require pharmacogenetic testing. And so if there's a drug with a very wide therapeutic margin used for a very short period of time, you might not care very much about predicting risk of efficacy or toxicity. But there are a couple of situations in which pharmacogenetics can really play a role. The first is when there's a narrow therapeutic index. And what that means is that there's a small range of doses at which the drug produces a beneficial effect without causing a severe drug reaction. And one example that I'll discuss later is the anticoagulant Warfarin. Pharmacogenetics can play a role when there is a differential response by race. And so when there are race effects on drug response, there tends to be, this tends to be a marker of some underlying genomic differences. Pharmacogenetics can also be used when there is a high variability in pharmacokinetics or pharmacodynamics of a drug, as well as situations in which an adverse drug reaction, the development of adverse drug reactions seems a bit idiosyncratic, where we don't really understand the mechanisms or reasons why they occur. In terms of the approach to pharmacogenetics, the studies I've listed here several study designs to really evaluate whether pharmacogenetics is useful. The gold standard is really to study in a randomized fashion genetically guided drug therapy against standard of care. It really offers the best level of evidence, but one of the barriers is that it first requires us to identify known pharmacogenetic determinants. And so sort of the first step. Randomized controlled trials can also be used to determine this. And so when we have pre-existing clinical trials, we can leverage them by using the DNA that's collected in clinical trials, if that was done, to associate with drug outcomes that were collected. And this can be an optimal design to really characterize pharmacogenetic determinants prior to evaluating whether there's an impact of gene-guided therapy versus standard care. We can also use population cohort studies to sample a population that might have been exposed to a given medication of interest and examine differences based on genotype. And finally, we can sample patients based on whether they experience an adverse event or have a poor treatment outcome, and look at the genetic predictors in the cases versus the controls. In terms of selecting genetic markers in a pharmacogenetic study, the initial studies were really hypothesis-driven, where specific signals were specific single variants or multiple genes that had some sort of biologic relevance were selected and tested based on potentially being well characterized to cause a drug response. And so essentially you're looking at genes in a particular drug pathway, transporter that might have an impact on your drug response. When there isn't really a clear understanding of the pharmacology of a drug, we can now use agnostic approaches using GWAS and high throughput platforms to evaluate for unsolved variability in drug response. And so that is more of a hypothesis generating approach rather than one that's targeted. Pharmacogenetics offers several advantages. The most obvious is the ability to perform patient stratification. And so genetic information can be used to define subgroups of individuals more or less likely to experience a response to the drug or develop side effects. And the point of this is to really avoid unnecessary treatment and maximize efficacy and giving the right medication to the right person to the right time at the right time. Pharmacogenetics also has the potential to optimize drug development, where we can potentially identify new drug targets that can lead to new therapies. And finally, drugs can be used to perturb the human organism. And so we can use a drug and apply it to an individual and measure the physiologic response to the drug. And if that response differs based on genetic variation at a particular locus, then we might understand a little bit more of the mode of action of the gene product and just understand a little bit more about underlying mechanism. And I'll provide some examples of that later in the talk. So I don't know if there are any questions at this point, but pause if anyone has any questions before moving on to some key examples of pharmacogenetic variation.
I think none right now, Dr. Li.
Okay, so let me move on. So the initial uses of pharmacogenetics were really mainly in cancer therapeutics, and the earliest successes were in targeted drug therapy. One example is the use of imatinib or Gleevec for chronic myeloid leukemia. And so give a little bit of background. Virtually all cases of CML or chronic myeloid leukemia are due to a genetic abnormality in the Philadelphia chromosome, which results from a reciprocal translocation between the long arms of chromosome 9 and 22. And so the result of this translocation is the production of this fusion protein, which is depicted in the slide. And this protein is a constitutively activated tyrosine kinase. And the drug imatinib was basically developed to competitively inhibit at the ATP binding site of the enzyme and causing inhibition of tyrosine phosphorylation. And so this slide illustrates a phase 1 trial in which 6 patients received imatinib and depicted, each line depicts the trend of the white blood cell count for a given individual. And what you can see here is within 2 weeks, the treatment led to a dramatic reduction in white blood cell count. And really, this is an example of the success in understanding the role that somatic mutation can play in disease and how a targeted therapy can be used to address this defect. And again here, other examples in oncology are listed regarding dysregulation of tyrosine kinases in cancer cells. One example is overexpression of a receptor tyrosine kinase in breast cancer, which has been subsequently been successfully targeted by a recombinant humanized monoclonal antibody against HER2. That medication is called Herceptin. And then there are other examples of small molecules used in non-small cell lung cancer to address defects in small deletions and point mutations in the EGFR. Drug metabolizing enzymes and transporters are also a major source of pharmacokinetic sources of variation. And so I'd like to provide 2 examples here. One is the story of CYP2C19 and Clopidogrel response, which is an example of how we can use this variant to understand efficacy. And then the other is an example of drug toxicity, which is the SLC01B1 gene variant in looking at statin-induced myopathy. So Clopidogrel is an antiplatelet agent. It's used in conjunction with aspirin to prevent ischemic events following acute coronary syndrome and percutaneous coronary intervention or stenting. Clopidogrel is a prodrug, which means that it requires conversion into an active metabolite by cytochrome P450 enzymes in the liver. And there are the genes encoding the CYP enzymes are polymorphic, and different alleles can cause reduced enzymatic function. And so people who carry reduced function polymorphisms have problems converting to the active metabolite, and this subsequently influences the degree of platelet inhibition on Clopidogrel. And so the hypothesis here is that individuals who have reduced function variants will have lower efficacy of Clopidogrel, and this could lead to higher rate of adverse cardiovascular outcomes. So this is a study that looked at 162 healthy subjects treated with Clopidogrel. And what's depicted here is based on their CYP2C19 genotypes. They've been characterized as having ultra rapid metabolizer status, ranging from that to poor metabolizers. And as can be seen in panel A, which is the pharmacokinetic response, measured as active metabolite concentrations, you can see that poor metabolizers had lower levels of active metabolite compared to those who were ultra-rapid metabolizers. Moreover, in panel B, which is the pharmacodynamic response in the same group of these healthy individuals who receive varying doses of Clopidogrel, panel B depicts the reduction in platelet aggregation across those different groups. And what you can see here again is that the carriers of the reduced function alleles had decreased platelet inhibition. And so these differences translate into clinical findings. And so this study looked at a clinical trial in subjects who were treated with Clopidogrel and had genetic data available to classify them as carriers or non-carriers. And they measured, based on the primary outcome of the study, this composite outcome of cardiovascular death from cardiovascular causes, myocardial infarction or stroke, and they found that carriers had a 1.5-fold increased risk of these events, as well as a 3.1-fold increased risk of a stent thrombosis or blockage of the stent that was placed and for which Clopidogrel is being used to keep it paid. And so, the question is whether this has been applied clinically given this data. And so in 2010, the FDA advised practitioners to consider alternative treatment in those who are poor metabolizers based on their CYP2C19 genotype, and to identify patients by genotyping who would fall into this category. And the question is, what has this really been taken up by the cardiology community? And currently, as it stands, genotyping is actually not currently recommended by the ACC and the AHA. And the reason cited is that there's a lack of prospective evidence. A lot of these studies are retrospective. Lack of prospective evidence looking at outcomes where individuals have been genotyped versus those who receive standard of care. The other wrinkle is that there are new antiplatelet inhibitors that are now available and often used and chosen over Clopidogrel. And so again, this finding hasn't really made it into mainstay clinical practice. Switching gears to another medication that's commonly used, statins, which are prescribed to lower low-density lipoprotein cholesterol and to prevent cardiovascular disease. They're very commonly prescribed, but one of the major reasons for non-adherence is the development of statin-induced myopathy, which can range from severe rhabdomyolysis or muscle damage, and then can be as mild as just some muscle aches. And so a genome-wide association study was conducted to search for genetic predictors of severe myopathy, conducted in 85 cases and 90 controls on 80 milligrams of simvastatin, which is a commonly used statin. And it identified a single strong association of myopathy with an intronic SNP located within SLCO1B1, which is a gene that codes for a hepatic transporter gene that regulates the uptake of statins. So the non-coding SNP that was illustrated in the Manhattan plot and the earlier slide was in nearly complete linkage disequilibrium with this non-synonymous SLP listed in this slide, where the polymorphism interferes with the localization of the transporter to the plasma membrane. And this results in higher blood concentrations of statins and is thought to be the mechanism by which individuals have a higher risk of statin side effects. And the odds ratio for this outcome was 4.5-fold increased risk per copy of the C allele. And it was relatively common where 25% of the general population tend to be carriers of this allele. So What's also interesting is that the risk of myopathy from this variant appears to be statin specific. And so in this figure, you can see that the percentage of individuals with adverse event is higher in those who carry the allele if they're on simvastatin, and lower with atorvastatin, and almost no difference by genotype looking at pravastatin. And so this is an opportunity basically to be able to use genotyping for this variant to inform statin selection and potentially impact adherence. And so there have been some randomized controlled trials that have studied genotype-informed statin therapy against usual care. And so one small study that I was involved in looked at 150 or so individuals where they were randomized and found that genotype-guided therapy showed some improvement in or higher rates of new statin prescriptions. So patients were more likely to take new statin prescriptions, providers were more likely to prescribe them. And then they had lower LDL at 3 months in those who received genotype-guided therapy. However, a larger study that was done more recently looking at about 400 individuals who were randomized to genotype-guided therapy versus control did not show differences in LDL cholesterol reductions. So these results, do not, results a little bit mixed, do not fully support kind of patient benefit from a standalone genotyping, but at the same time, we didn't see any sort of harms to performing this. And so that certainly reduces concerns from those who are worried about the use of genetic information in clinical context.
Dr. Li, there's a hand up from Monty Krieger. I think we can unmute you to let Dr. Krieger speak.
Thanks. Thank you for this, Josephine. I just wanted to ask you two things. One, recently there have been studies suggesting that a large fraction of the complaints about myopathy and aches are in fact not due to the statin itself. Controls with placebo have shown the same, almost the same rate, suggesting that that maybe that's why you don't see such large effects in other studies. Are you familiar with those?
I haven't looked into that specifically, but I certainly
only the last week or couple weeks that's come out, that might be worth looking at. And the other is, I was under the impression that with the Clopidogrel that a number of studies with the CYP variants, in fact, later on did not show clinical effects. Is that the case?
Right. So there are some. I was showing some of the initial excitement around the variant, but there are other studies looking at randomizing individuals to because the one I showed was kind of looking at a randomized clinical trial that was conducted, and then the genotype data was used to kind of look at those outcomes. But prospective studies looking at genotype guided therapy using the variant. The genetic information versus usual care did not really show much of a difference. So that,
yeah, I mean, that to me, that was always incredibly frustrating because it all makes sense that it should.
Absolutely.
All the data with the processing of the drug and everything else, yet it didn't seem to have a clinical effect. Do you know if there's any kind of consensus in the field as to why that ended up that way?
I know there's we're looking into it. The one study I do remember seeing recently published maybe a year or two ago looking at that did say that the outcomes, the pre-specified endpoints weren't met, but there were some sort of secondary endpoints that there might have been a small difference, but not to the point that was sort of initially preset by the study based on their power and whatnot. So yeah, to be determined. Okay, so moving on. So we talked about transporters and enzymes involved in kind of drug metabolism. But I also want to move to some pharmacodynamic sources of variation. And the key example here is with warfarin dosing. So warfarin is an anticoagulant with a narrow therapeutic window, as I mentioned, where the daily dose is depicted in this figure as highly variable among individuals. And so warfarin achieves its anticoagulant effect through inhibition of VCOR C1, which catalyzes the conversion of vitamin K epoxide to the active reduced form of vitamin K. And VCOR C1 variation has been associated with inter-individual dosing variability and clinical outcomes. And so here I present a study looking at 10 common VCOR C1 SNPs and that were then organized into haplotype groups. So they identified a low dose haplotype, which is A, and a high dose haplotype, which is B. And patients in the population, the study population, were genotyped and assigned haplotype combination based on the results. And so the figure on the left here shows that at the ones who were AA had a much lower warfarin maintenance dose compared to those who were AB, and then compared to those who were BB. And then another study looked at clinical outcomes. And so they measured the time to the first therapeutic INR. And INR is basically a blood test that measures how thin the blood is on warfarin. And they found that those who belong to the low dose haplotype group reached this therapeutic INR much quicker. So they had a decreased time to the first INR. And then they also looked at those who had an INR that was greater than 4, so beyond what is targeted and would increase the risk of bleeding events. And those in the low dose haplotype group also had a decreased time to that as well. And again, kind of similar to the Clopidogrel story, you sort of wonder, the application of this in the clinical setting. And there's, again, in 2007, the FDA updated labeling for warfarin, stating that lower initiation doses should be considered for those with certain genetic variants. And then subsequent trials looking at genotype-guided warfarin dosing have been sort of mixed. And then this is a common theme as well, which is that there are new therapies available, the factor 10A inhibitors, and these have been more commonly used as opposed to warfarin nowadays, sort of causing some of these findings to be less relevant in the clinical setting. Final kind of source of pharmacogenetic variation I want to discuss is maybe found in genetic variation linked to the pathophysiology of the underlying disease. And so studying genetic variants that relate to disease risk may help prioritize why they might influence drug response. So here, switching gears to hepatitis C, years and years ago, the treatment for hepatitis C, the standard of care was pegylated interferon ribavirin therapy, which is no longer the standard of care now, but at the time was, and was very poorly tolerated due to side effects. People felt very ill, like they were having the flu. Researchers were interested in identifying genetic predictors of treatment response as measured by sustained virological response, which is the absence of detectable virus at the end of follow-up. And they identified this gene variant near the IL-28B gene, which encodes interferon gamma-3. And here we see that on the left, the percentage of individuals who reached sustained virologic response differed by IL-28B genotype, where the C allele was the response allele. And what's interesting here is that the frequency of the C allele, when you stratify by ancestry, was much lower in African Americans. And in the second panel, you can see that it somewhat correlates to the proportion of sustained virologic response. And so there might be some explanation of ethnic-specific effects here, and illustrates why it's important to think about pharmacogenomics applying to the whole population and to study very different ethnicities because the response is different. And then the thought through these studies was that hepatitis C infection can stimulate endogenous interferon production, and that those with the responder genotype may have a lower endogenous response to the virus. And so sheds a little bit of light into pathophysiology of the disease. However, again, common theme is that this, as I mentioned at the beginning, interferon therapy is no longer the mainstay therapy of hepatitis C, and that there are new antiviral regimens that are much more tolerable, and it makes these genetic findings much less relevant. Okay, I can stop for additional questions or comments before moving on to type 2 diabetes. I don't know if there are any comments in the chat or.
Dr. Li, I actually just had one question for you before you move on, if that's okay. Sure. When looking at all of those studies, many times they had to set a dose cutoff for looking for a response to detect their a phenotype. And I was wondering if you have any insights into how sometimes those dose thresholds were chosen, and whether you think that that decision impacted the ability to detect a true association or not.
I think a lot of the studies were basically using doses that were used in clinical practice. Certainly the GWAS and statin, pharmacogenetics was using a relatively higher dose of simvastatin to identify these cases. But I think it sort of depends on what the platform is to study. The pharmacogenetic studies that are using existing data are sort of piggybacking off of what was done for the original study, but the ones evaluating genotype-guided therapy will use doses that are used in routine practice. Okay, so I'll move on to application in type 2 diabetes, but before I do that, I wanted to highlight that pharmacogenetics has been successful in neonatal diabetes, and kind of describe this. I've presented here a figure showing insulin secretion pathway in the beta cell in response to glucose. And so panel B here illustrates glucose metabolism, where presence of glucose causes increased intracellular levels of ATP, leading to closure of potassium, the ATP-sensitive potassium channel, which is a key regulator of beta cell insulin secretion. And so mutations in this channel result in a failure of the channel to close, resulting in impaired insulin secretion and hyperglycemia. And so what was discovered is that heterozygous activating mutations in KCNJ11, which encodes a subunit of this potassium, ATP-sensitive potassium channel, can cause 30 to 60% of cases of diabetes diagnosed in patients under 6 months of age. And these patients are often presenting with severe hyperglycemia or ketoacidosis and are treated with insulin. And so what's been really interesting in these activating mutations is that they can be treated with a sulfonylurea. And so sulfonylurea is an anti-diabetes medication that closes the ATP channel by ATP-independent route. And so sulfonylureas have been shown to be effectively used in place of insulin for treating neonatal diabetes caused by these activating mutations. And so here we illustrate, this panel illustrates individuals that were on insulin initially and then transitioned to a sulfonylurea and had improvement of their hemoglobin A1C. And it's very remarkable that these individuals were able to come off of an insulin injection and be treated with an oral agent due to our understanding of the underlying mechanism. Pharmacogenetics has been studied in metformin, which is the first-line agent for type 2 diabetes. Metformin is the first line because it's safe, effective, and has a low cost. It's inexpensive. But despite its widespread use, the mechanism isn't entirely clear. Metformin also doesn't work for all individuals. We know that from clinical studies that 21% of patients fail metformin the first 5 years of treatment. And so this represents a group that is of interest. And the failure rate can be quite high in youth with newly diagnosed type 2 diabetes, which is a group in which diabetes incidence is rising rapidly. And so there is a need to capture and describe the full range of pharmacologic responses for metformin and identify those who might not benefit due to inadequate achievement of glycemic targets or due to having side effects. And part of the challenge here is that we don't quite understand the molecular target of metformin and a precise mechanism of action. And so candidate genes have been, gene studies have been done to evaluate for predictors of metformin response, as well as genome-wide association studies. And so this is a GWAS meta-analysis of 10,000 participants with harmonized measure of metformin response. So looking at change in hemoglobin A1C. And this variant in the GLUT2 transporter gene, so an intron at the SLC2A2 gene, which encodes the GLUT2 glucose transporter, was found to have an association with a greater reduction in HbA1c compared to the T homozygotes. And so carriers who of the variant were found to have greater effect on metformin, and that effect equated to a dose of 550 milligrams of metformin. Interestingly, there's support for biological relevance where the same variant influenced GLUT2 expression in the liver as well. Pharmacogenetics can also be used to understand the function of identified gene variants. And so this polymorphism listed in TCF7L2 has been the strongest association with T2 risk discovered from GWAS, but the precise mechanism is unclear. There's thought that the variant diminishes beta cell function, but there have been many efforts to identify for other contributing mechanisms. And so as described earlier, we can use drugs to perturb the human organism and help understand modes of action of variants. And so this was the basis for a study that was conducted Jose Flores' lab, Sugar MGH, the study to understand genetics of the acute response to metformin and glipizide in humans. And in this study, 1,000 healthy participants at risk of type 2 diabetes were given glipizide challenge of 5 milligrams one-time dose, and subsequent measurements of glucose and insulin were performed over 240 minutes. And then they received a metformin challenge for 4 doses. And then received an oral glucose tolerance test on metformin to understand what happens to insulin resistance and insulin sensitivity after metformin. And so the 2 drugs were selected to probe the 2 different limbs of the glucose homeostatic system. And based on how an individual responds by variant, we can understand a little bit more about underlying mechanism. And so here, we found that the T allele, TCF7L2 variant, was associated with no major changes in glucose or insulin, but higher fasting total and active GLP-1 levels. And GLP-1 is an incretin or gut hormone that is released after eating and augments the secretion of insulin from the pancreatic beta cells through a glucose-dependent mechanism. And so what's interesting is that while there were no major changes in glucose or insulin by genotype, we do see an increase in incretin levels, which is somewhat suggestive of some incretin resistance in individuals with the who carry the T allele as a potential mechanism by which TCF7L2 affects type 2 diabetes risk. We can also now, the field is moving more towards polygenic risk scores, where we can aggregate many variants across the genome, which can provide increased power because individual variants only have a modest effect. And so in our cohort, we looked at polygenic scores comprised of genome-wide significant loci for type 2 diabetes risk and tested against drug response outcomes. And the thought is that people who have an increased risk have most to gain from therapy and might postulate that they might notice a difference by genotype there. And so here we looked at the glipizide challenge, so sort of that first drug perturbation I mentioned, where individuals were given glipizide, which is a sulfonylurea that causes insulin secretion and drop in glucose. And so here across the cohort, you can see that the glucose drops over time, lowest at 120 minutes, and insulin peaks at 60 minutes. And so from these, we designed multiple phenotypes of glipizide response and associated polygenic score with markers of the response. And so what we found was that those who had a higher burden of type 2 diabetes genetic variants or higher risk based on their genotypes were associated with a more robust response to glipizide, and that was characterized by a greater cumulative drop in glucose over the course of the challenge, a shorter time to the lowest glucose that was achieved, a shorter time to the insulin peak that was achieved as a result of the glipizide, and a steeper slope to the lowest glucose. And all of these are consistent with a more robust response. We also looked at a longitudinal cohort of individuals with established type 2 diabetes and who had received a sulfonylurea. We tested for association with reduction in hemoglobin A1C at 1 year. And we confirmed our finding in that for every standard deviation increase in the polygenic score, individuals had a greater A1C reduction. And we looked also at the extremes. So we compared the top with the bottom decile of the distribution of polygenic score. And we found that the difference was much greater when comparing the tails at the end of the distribution. And this may suggest that, again, the use of polygenic scores in pharmacogenetics at those who are at the extremes, as they maybe have the most to sort of gain from this. Okay. I'm going to switch gears a little bit and talk about some resources for those who might be interested in implementation of pharmacogenetics and just understanding the body of literature for a particular disease or particular drug. I want to mention that the FDA last year actually posted a table of over 50 gene-drug interactions supported by scientific evidence. This is a small fraction of the list, but it's limited really to pharmacogenetic associations related to drug metabolizing enzyme gene variants, drug transporter gene variants, and some related to adverse events. So, and this list will continue to be modified and updated as new evidence arises. There are also several resources that facilitate the translation of findings into sort of actionable recommendations for various drugs. And so the Clinical Pharmacogenetics Implementation Consortium, or CPIC, has authored several publications across many drugs that tried to provide some guidance on how to use genetic information in selecting drugs and selecting doses. And this is one such example, looking at a HLA genotype and use of carbamazepine, where there is a concern for cutaneous side effects.
Dr. Li, there were a couple of questions about your polygenic risk score for type 2 diabetes. People are very interested. Dr. Lewis was wondering if you could put your percent decrease for one standard deviation increase in the polygenic score in the context of clinical measures.
Let me go back to that slide here. assuming you're talking about this particular one. So, we do measure hemoglobin A1C reduction as a response to anti-diabetes medications. And so that standard deviation decrease is quite small, and that's not very clinically meaningful, to be honest. And that's why we chose to examine those at the extremes. And so the most modest glucose lowering agents produce sort of effective 0.5%, whereas other agents such as metformin are greater 1.5%. And so that's sort of in comparison to what we're seeing here with the extremes, which is 0.27%.
Uh-huh. And there was another question where Dr. Henigberg was wondering if you've looked at this with any other classes of antihyperglycemic medications, or if you think this is unique or specific to sulfonylureas.
Yeah, that's a great question. So we did look at metformin for the T2D risk score, and we did not see an impact in for the acute metformin response in our cohort sugar MGH. And we are certainly very interested in looking at the use of scores to predict drug response for other classes, and have designed studies to pursue that.
Great. And then, sorry, there was one question who I think we've unmuted Megan to be able to ask. Megan, I think you're, yeah, perfect.
That was an error. I accidentally, sorry, I didn't have a question. I apologize.
Oh, no problem. Thanks so much. Sorry, thank you, Dr. Li.
Great, no problem. Yeah. Advance. Okay. And then the final resource I wanted to discuss here is PharmGKB, which is an online resource that can access. I should have put the link there. But it basically is a team that collects, curates, and disseminates information about the body of knowledge on various drug responses. And so they have drug label annotations from the FDA and other medicine agencies. They include the guidelines such as the CPIC ones I had mentioned. They have diagrams, some of which I showed earlier in this talk, that kind of depict the pharmacokinetics and pharmacodynamics of a given drug with relevant pharmacogenetic associations. And then you can actually search and look at what literature has been published on some of these patients feature. Okay. And so in the last 10 minutes or so, I want to discuss challenges and future directions in this field. And so there are a couple of barriers to implementation, some of which sort of have been brought up throughout the talk and also by others in their questions. But one major theme is that discovery in pharmacogenetics really lags behind the development of new therapies. And so what we keep seeing is that there are new agents that are arising and old ones being abandoned, and that we need to really keep up with the pace of pharmacogenetics. And one problem is also that in sort of agnostic genome-wide association studies, some of the findings tend to have small effect sizes, and it's unclear if they're clinically meaningful. And one way to circumvent this, which is actively being studied, is the use of polygenic scores when you can aggregate many variants across the genome. I presented the use of a polygenic score for kind of disease risk, but you can imagine you can create scores for drug response and test those as well. There's some challenges with interpreting complex results. We need to also make sure that our results are applicable across all ethnicity groups. And so this really calls for, multi-ethnic cohorts and as gene frequencies can really vary across ethnicities. We have also, a lack of prospective RCTs to really address whether they improve testing improve patient outcomes. We need to understand whether, performing these tests are really cost effective. And then finally, we need to have pharmacogenic testing really be accepted by patients and clinicians in order to really implement them in clinical setting. And so where do I sort of see the future of pharmacogenetics? What kind of improvements or areas do we really need to venture into. I think, as I mentioned, we need large prospective studies. We need to be doing routine data collection and studies so that we can have large cohorts to really measure effects and detect them. We need evidence base to support implementation, and some of the variants that are selected to be carried forward into clinical use need to be well validated. They need to be common enough that it's worth measuring in the general population, and then we need guidelines and interpreting test results and experts to help guide that. We also need to, we didn't really talk about this too much in this talk, but there needs to be infrastructure in the healthcare system and the medical record system that allows the integration of genetic information. One kind of new area that many are interested in is the use of large biobanks to really gather genetic data, and whether that information in the biobank can be used to sort of prospectively make a decision is something that is of the future. And then finally, we need to educate and train experts in this field. And so, for pharmacogenetics is here, it's been here, and I think there are many opportunities for advancing precision medicine, developing new drugs, and understanding new genetic discovery and underlying mechanism. Extending to treatments for complex disease like type 2 diabetes could certainly pose challenges for a polygenic disease versus looking specifically at a transporter or a particular adverse event. We need to clearly demonstrate impact on clinical outcomes, which has been a challenge thus far. And the future may involve preemptive genetic testing now that the cost of testing genotyping has gone way down, but doing it only once in an individual, streamlining those results so that all actionable variants are all done at the same time early in life and can be used many years prospectively. And with that, that's the end of my talk. I'd like to thank all of you for attending, the Broad MPG community for inviting me, and to my mentor, Jose Flores, and members of the Flores Lab.
Great, thank you so much, Dr. Li. That was such a wonderful talk. Actually, Dr. Flores has a comment / question that I was going to read out. It seems to me that to maximize the utility of the genetic information in pharmacogenomic decisions, one has to take the cost variable out of the equation. The conversation would be very different if instead of having to order an expensive genotyping test in advance of a therapeutic decision, the genetic information was present so in everyone as soon as they enter a healthcare system genome-wide, so it could be applied for as clinically indicated. And the decision flows based on all this information rather than including an extra costly step in decision making. So it sounds like exactly what you said on your
Yeah, absolutely. Just having that available for everybody, and so I wonder if now that we're collecting all these information in biobanks, this is an opportunity for that to facilitate the use of pharmacogenetic data.
Yeah, and then there was a follow-up question to that in the chat, which was whether anyone had studied return of results to patients and their primary care physicians with regard to pharmacogenomics in people who have WGS or other clinical or research tests, and whether doctors and patients switch medications based on those results.
So the statin study I mentioned, one of them was the results were returned to both the patients and the providers, and they measured sort of some kind of soft survey-based measures of sort of how they viewed their medications based off of receiving that information. And so there have been some initial studies kind of looking at both the response and reception to that information. Certainly, there can be barriers in utilizing this sort of in a clinical setting for those who don't feel comfortable or don't feel like they have the proper support and guidance to do that.
And then there was one last question before we'll break for the next session, but Dr. Krieger was wondering if you thought genotyping should be required for phase 2 and 3 studies of all drugs.
Yeah, I mean, I think that's an excellent suggestion. I think you know, in our work, we've for some of the newer anti-diabetes agents, you know, there isn't genotyping done, and so it does limit the ability to sort of compile large scale, large studies to be able to power genetic analyses. And so I think including DNA collection would really help facilitate that.